show you the images. And I was going to draw on the iPad because it's easier, but then I forgot and I logged into the meeting on the computer. I don't think I can change without losing all of you. So just going to have to go with it. So I'm going to put myself back over here. And I'm probably just going to use the whiteboard and use the markers. So today we need to cover a bunch of things still. We need to cover gene regulation or genetic control, as well as horizontal gene transfer and mutations. So let's get started. Make sure, yep, it is recording. Okay, so the first thing we're going to talk about real briefly is gene regulation. And we're going to discuss this, oh, excuse me. We're going to discuss this in terms of the what's called the lac operon. Because this is a simple example of how a gene can be regulated or turned on and off. And the point of this is to show you how it can work. And of course, in humans, it's going to be much more complicated than this. But we're going to show you a simple gene regulation in bacteria. And this gene regulation focuses on transcription and translation. And that is where what we would call transcriptional control. And this is where many genes are regulated, because if it's, especially in bacteria, if it's transcribed, it's going to be translated. There's no break or post-transcriptional modifications like in eukaryotes. So we're going to discuss this in terms of the lac operon, and why this is so important is for a couple of reasons, of course, because in bacteria, we want to be able to regulate genes because we might be able to use that to kill the bacteria as a therapeutic agent. In humans, we want to know why a gene turns on or why it turns off, why it's on for part of our lives, why it's off for the rest of our lives. Think about those of you that have already had anatomy and physiology. Think about hemoglobin, for example. We have fetal hemoglobin when we're in the uterus, and then as soon as we're born, we switch to making adult hemoglobin. And that fetal hemoglobin gene never gets turned on again. And somehow the adult hemoglobin gene does get turned on, and that would be some sort of transcriptional control. So anyway, oh, excuse me, I'm going to have to apologize. But closing up the house, apparently there's a lot of dust in my house because I don't clean very often. Anyway, all right, so the lac operon. So we'll start with what's an operon. An operon is a group of genes that's controlled by one regulatory region. I like to say with one switch, meaning one way to turn it on and turn it off. In the case of the lac operon, lac does stand for lactose. And in the case of the lac operon, we're talking the genes that are being controlled are actually, believe it or not, they're called Z, Y, and A are the genes. And you'll see those referred to as the structural genes, because they're actually going to, they produce enzymes, each one of them. And those enzymes are involved with the transport and digesting or catabolism of lactose. This is not how lactose intolerance works in humans. So someone always asks, but that's not how it works. Hang on, guys, I have to let somebody else in. Okay, so I usually draw the whole setup so it looks like this. So can you mute yourself, please? Thanks. So the control region is at the beginning. So this is, so let's say we're inside the bacteria here, right? And here's the DNA. This lac operon, it actually occurs pretty early, like up here, right? This is just one piece of the genome of the bacteria. And in this case, it actually is our favorite laboratory animal of microbiology. It is E. coli. So the three genes at the front that are all involved in the switch are called the I gene, P, IPO, and then we have the three genes themselves. C, Y, and A. Y gene produces a protein, so it has its own promoter region. And RNA, remember, for transcription, we're going to need RNA polymerase. All right. I gene has its own internal promoter. RNA polymerase is going to bind to that promoter and produce the protein. So first produce the messenger RNA for the I gene. And then that is going to produce a protein. And I'm going to make that protein purple. Actually, look to you. Make him orange. That produces a protein, and it actually does kind of look like a horseshoe. The protein's name is called repressor protein because it represses or keeps the operon turned off. So this is a constitutive, we say, gene, meaning it's on all the time. And this protein is actually called repressor protein. I know, why didn't they call it R? They just didn't. It's called the I gene. So repressor protein actually binds here to the operator. So I'm going to try to draw it without ruining my tip of my orange marker here and making it turn all black, because I'll never get it out of there, and then orange won't look orange anymore. So we're going to do this. Right? Because I'm trying to show you that repressor protein, when it sits on operator, it's like a clamp. It sits over the top like this. Okay. When repressor protein is repressing, the operon is off. So what I've just drawn here is the off position. And the operon is off when there is plenty of glucose and the bacteria is using aerobic respiration or fermentation or whatever it's doing, or anaerobic respiration, in order to produce plenty of ATP. But when there's plenty of glucose, we don't need to turn this on. Try to remember that every time you make a protein, and we didn't particularly cover this, but for translation, every time you add one of those amino acids, it costs about 2 ATP. So if we have to keep making proteins over and over again, it gets very expensive. So the lac operon is off. We are not digesting and transporting lactose when there is plenty of glucose. However, this needs to be turned on when? It needs to be turned on when the level of glucose drops. Right, so I put a down arrow, but I'll also write drops just in case the down arrow is not self-explanatory. Also, when there is a lower concentration of ATP, and at the same time, a higher concentration of cyclic AMP. So cyclic AMP is a warning signal to cells that they need to make more ATP. And so it's a warning signal goes out and the cells sort of jump to trying to find a way to make more ATP. However, this also will only come on when lactose is present. If there's no lactose in the food source, in the media, this isn't going to work. Nothing happens. You got to have lactose. The lac operon, we call it, your book you'll see it called an inducible operon. That means it is turned on or induced when lactose is present. So I'm going to make the lactose be these, usually make it these little blue circles. So if I can find a blue that works. And so these blue dots are lactose. Lactose. Yeah. And how this works is pretty simple. Repressor protein likes operator, kind of, but it is in love with lactose. It has a binding affinity, meaning it binds better and more quickly and binds longer to lactose, not to operator. So when lactose is present, what's going to happen is I'm going to redraw my operon. And I realize it's kind of tight here, but I'm just going to redraw for the on position. So I'm just going to make I, P, and O a little bit smaller. when lactose is present, when this operon is on, I is still making repressor protein. However, repressor protein, instead of binding to operator at this point, it is going to be bound to lactose. I'm going to try to draw some little blue circles again inside of here. Well, if it's not bound to operator, then our friend RNA polymerase can bind to promoter. I'm going to put RNA polymerase here. And once RNA polymerase binds to promoter and there's no block at operator, it is going to transcribe. So I'll make 3 messages and I'll label those messenger RNA. It is going to, I'll put an arrow here, like RNA polymerase is going to sit down, recognize the promoter, it's probably a Tata box, and it's going to make messenger RNA transcripts for Z, Y, and A, which will then be translated in ribosomes into, and I'm just going to draw 3 blobby proteins because remember they're enzymes, into the enzymes, their real names you don't have to know, don't worry about it. Z, Y, and A, which remember transport and digest lactose. Awesome. Now the operon is on, the cell can increase its ATP production. Yay, happy yellow sun. Right? Increase its ATP production, and this is going to continue until the cell runs out of lactose, and then if the cell runs out of lactose, it will then go back to the off position, because when there's no more lactose, when these last lactose molecules have been pulled out of repressor protein and used to make ATP, repressor is going to go back into the operator and shut it off again. So I'm going to pause for a second. Immediate questions about the lac operon before I go on, because I am going to have to erase this to continue with the next little section that we need to talk about. I can actually, I think I'm going to, yeah, fine. Questions, comments, worries about that? You will not have to be able to discuss the trip operon, which is another operon, which is not an inducible operon, it's actually a repressible operon. So in that case, the end product actually goes back, I'm trying to put my head next to the ATP so you can see it. In that case, the end product goes back and shuts off the operon. But you can skip, and skip means not going to be on the test. You can skip the trip operon, which comes second, comes after this in your textbook. All right, so I'm going to erase this. Turn my page here. Erase this. This is figure 918. And then we're going to move on to talk about horizontal gene transfer. Go fast here. All right, so the next thing we need to talk about is horizontal gene transfer, which means just like it sounds. Remember, bacteria do not do mitosis, so they do not do sexual reproduction. Horizontal. But what they can do is they can give DNA, particularly DNA that's in a plasmid, they can give it to other neighboring cells. Now, there are 4 mechanisms or 4 major ways that bacteria can transfer DNA from one cell to another. And I'm just going to list them real fast, because I'm going to do them in a different order than in your book. Transduction, conjugation, transformation, and transposons. Come to genetics, learn more about transposons. For transposons, you only need a definition only. Definition. Sorry, but yeah, I'm just like your parents. I can't text and talk at the I can't text and think at the same time. I have to actually like talk while I'm spelling it out. So this is definition only for you. Come to genetics, learn more about it. We're going to spend the rest of our horizontal gene transfer conversations talking about these three things: transduction, conjugation, and transformation. I like to start with transduction because it's historical and it's related to actually things you learned about in chapter 9. And it is actually a big part of the story of how scientists figured out that DNA was the genetic material, not protein or RNA for the most part. So we're going to start with, we're going to start with transformation. What am I doing here? 1, 2. For transduction, you guys only need to know, why am I having this mental block? You only need to know generalized transduction, not specialized transduction. Sorry, having a minute there. We're going to start with transformation. So usually we define transformation as the transfer of naked DNA, and it's usually pieces of DNA. And what we mean by naked is it's not enclosed in a protein coat or a phospholipid bilayer membrane. This occurs only when one cell dies and a neighbor takes in its DNA. So here's a bacteria, and here's its little chromosome, like I like to draw it. And when this cell dies, actually this isn't going to be E. coli this time, it's going to be Streptococcus pneumoniae. When the cell actually dies and the DNA is fragmented into pieces, little bits like this, so not a whole circular chromosome, but little pieces, a neighboring DNA, a neighboring cell can say, oh, I'm going to take that in and add it to my genome. So I'll make that cell's genome a different color. Let's utilize this orange. So this bacteria, its DNA was going to be the orange, and it's going to take in a little piece of the blue DNA, and it's going to become part of its chromosome. That's transformation. What caused this cell to die? Well, maybe it was heated, right? Maybe heat killed it, or maybe it was treated with some antibiotics, right? For whatever reason, cell number 1 dies, and its DNA, that's this DNA from cell number 1, is picked up by cell number 2 and becomes part of their chromosome. Why do we care? Well, we care because sometimes doing this transformation, you can actually move genes that cell number 1 had into cell number 2, and those are genes that cell number 2 did not have. That's what we call horizontal gene transfer, because we're moving DNA from one cell to another. This experiment was actually conducted by a man named Griffith in, I believe it was 1918. And he wasn't studying E. coli, he was studying Streptococcus pneumoniae. So these would be round bacteria, not the E. coli shape. And he was studying Streptococcus pneumoniae that was actually causing pneumonia, which is why it's called Streptococcus pneumoniae. Which, and remember, Streptococcus are going to be round coccus, spheres, strepto in chains. And what he found was he had 2 different strains of the bacteria, 2 different strains. One that could form capsules, and one without capsules, without. And what he determined was the one with the capsules, that's the one that gave you the pneumonia that most people died from. But if he mixed together dead bacteria that had the gene for capsules with live bacteria that did not, he could transform the previously no capsule bacteria into capsule bacteria, and they would cause pneumonia and death. Pay attention to the year, 1918. There aren't any antibiotics yet, right? So pneumonia was often a fatal disease, right? Because there wasn't much treatment except supportive care. In the real world, how often does this happen? With pretty fair amount of regularity. In the laboratory, in order to make this occur, like in a molecular biology laboratory, we sometimes use transformation. However, we treat these cells, the second cells, we treat them with a chemical, it's just calcium chloride, or with electricity. We give them little electric shocks to open up their membranes so that they will, their outer membranes, so that they'll take in the DNA. They'll be more likely to take the DNA. Probably what is much more common is the second thing here on my list. It's still on the second thing, it's in the middle, conjugation. The details of conjugation, I'm not going to ask you about HFR cells. I know your book goes into all of that. We're just going to talk about F+ cells and F- cells for conjugation. So conjugation has to do with when the bacteria, and I'm going to switch drawing the bacteria from horizontal to vertical just to save myself some board space because I don't have much space here. We're starting with a cell that has a plasmid. So I'm going to draw this bacteria and give it this enlarged bit of DNA here. So that's its regular chromosome, right, in the nuclear area. And then the cell also has, let's use the, can you see the difference between the green? Yeah, you can see that's different. So this is the plasmid. Plasmids on them, remember, they have genes in order to make those pili or sex pilus, a pilus, singular, pili, sometimes more than one, in order to transfer the genes, a copy of this whole plasmid to a neighboring cell. So this cell that has the plasmid, we refer to them as the F+ cell or the donor cell because they're going to donate the plasmid to their neighbor here who does not have the plasmid. So this cell starts out as an F- or as a recipient cell. And this second cell here only has the chromosome and not the plasmid. Okay. And I will redraw for you. What happens is our F+ cell is going to make some protein. So it's going to have to transcribe and translate so we need RNA polymerase for that. I'm going to transcribe and then translate a copy. Doesn't give it away. It makes a copy of the plasmid, sends it across the bridge, and now our formerly F- cell is going to become an F+ cell or a donor cell once it receives a copy of the plasmid. Again, why is this important? Well, many times on the plasmid, remember, get rid of this stuff about Streptococcus here. Remember, plasmids contain bonus DNA. So it's not essential, but if you have it, you're going to be much more successful at causing an infection. So it's good for the bacteria, bad for the host, like if we were the host of this infection. Non-essential, but usually virulence genes. And virulence those are factors that increase the likelihood that you're going to cause an infection. For example, the ability to make a toxin. That'd be good for the bacteria, bad for me. Good for the bacteria, bad for the host, right? The ability to have antibiotic resistance. So I make an enzyme, let's say, the bacteria on the plasmid, there's an enzyme that's going to allow me to inactivate penicillin. Ooh, good for the bacteria, bad for the host, right? Those are some common things that you would find on plasmids. And then the last method is transduction, which I incorrectly put first. And for transduction, it'll make more sense why we call transformation naked DNA when I talk about transduction. Transduction involves a virus. I know we haven't done viruses yet. We're going to do those on Wednesday. So transduction, a virus that is specific for bacteria. They have a special name. They're called bacteriophage, but most people just call them phage because bacteriophage can be quite a mouthful. So I'm going to get rid of these other things here so I can talk, so I have a little more space to talk about transduction. So for transduction, for now, you just need to know about what we call generalized transduction. On Wednesday, we're going to talk about bacteriophage, and I will teach you then about specialized transduction. But for today, we're just going to talk about generalized transduction. And how this works is, this is the virus that is going to infect the bacteria. And so here's our bacteria, living its bacteria life, right? And what can happen with generalized, so the virus has its own DNA that it's going to inject into the cell, and it's going to literally take over this cell and produce, so I'll just do this real quickly. Viral DNA takes over the bacteria and synthesizes inside of it a whole phage army. So I'm just going to draw lots of these little bacteriophages. And I don't know why, but what always sticks in my mind is the Spy Kids movie, the first one where he was making that little robot army of the children, right? And what happens is most of these phages get phage DNA put inside of them. But when the phage first enters the cell, in order to take over, it has to chop up the host DNA into little bits. And occasionally, some of the host DNA gets accidentally packaged into the phage. The rest of it did not. It's still laying over here, all defunct. When this phage, only this one, when this one infects the next cell, you see how it's bringing blue DNA, which is the same as the host DNA? So it's bringing new DNA that can be added to this host chromosome. And for right now, that's all you need to know about transduction and generalized transduction. As I mentioned, we'll talk about specialized transduction later, Wednesday, when we talk about 6. And the last thing we need to cover today is about mutations. I'm going to erase this. And we'll talk about mutations and how to test for mutations. Some of the colors here, they don't erase as easily. All right, last topic, topic 3 for today, mutations. So a mutation is any change to the DNA. Output to the DNA sequence. Now, that can be spontaneous. That means it's a mistake by DNA polymerase that is not corrected by DNA polymerase 1. I'll just put DNA polymerase mistake that is not corrected. The spontaneous mutation rate has been measured, particularly in bacteria because they multiply so quickly. It occurs about 1 in 1 million bases, which translates to about 1 in 1,000 genes. It's not about, that's why I put the squiggly there, right? Approximately 1 in 1,000 genes. So if we think about this, E. coli has about 4,000 genes. So every time the cell divides, which of course is about every 20 minutes, we're going to have 4 spontaneous mutations. Okay. The spontaneous mutation rate, if it's increased because of a mutagen, right? So that is either a physical or a chemical that increases the spontaneous mutation rate. Okay. Physical, that means radiation, basically, of any type. And chemical mutagens, of course, those would be anything that can be naturally occurring or man-made that, again, increase the spontaneous mutation rate. The effects of a mutagen or mutation fall into a few categories. And actually, before we even talk about the effects, we're going to limit ourselves to point mutations here. A point mutation is a single base change. Most often it is what we call a base substitution, meaning one base is substituted for another, and it is most likely purine for purine, pyrimidine for pyrimidine. So if you think about that, that means A gets changed accidentally, a G gets put in, or if it's a T that's supposed to be there accidentally, a C gets placed in. And then that's going to change the DNA, which potentially could change the RNA, which could potentially change the protein once that RNA is transcribed and translated. All right, now they fall into some basic categories. A silent. So that's when you have a base change. I mean, is it from chemistry? Is it okay if I just use a triangle to represent change? I'll put it over here. Okay. When you have a base change to the DNA, right, that's going to cause a base change to the RNA, the messenger RNA. But, and this is an important but, no change to the amino acid that's coded for. So it's a silent mutation. If you look at the chart, which I can't show you right now because I don't have it, but if you look at the dictionary or that chart that we talked about last time with the messenger RNAs and how they produce, with the anticodon, how they produce the amino acid chain, you'll notice that some of those codes, I think of serine particularly, or actually it's proline, I think. CC anything codes for proline in the message. So that would be a good example of a silent mutation, right? So if the DNA, the coding or the template strand of DNA said GGG, and that got changed to GGA, right? Purine for purine, right? The original messenger RNA would say CCC, which I'm pretty sure is proline, right? The mutated RNA, mutated messenger RNA, right? Instead of being CCC, it would be CCU, but that codes for the same amino acid. So it's silent because we still get the same amino acid, so the protein is okay. I'm gonna just check that real fast to make sure that that's an accurate example. Yes, CC anything codes for proline. Sorry. So that would be a good example of a silent mutation. Now, if there's a base substitution, that is going to cause a change to the message that changes the amino acid, then we refer to that as a missense mutation. So I'm going to erase this, and I'm going to erase all of this. I need more space for the frame shift. So the second sort of category, so the first one is a silent, right? Second sort of category is what we call a missense mutation. And that's defined as a change to the DNA that causes a change to the messenger RNA that results in different A period A for amino acid, a different amino acid in the protein. Might be a problem, might not be a problem. Depends on the protein and depends on the amino acid. If it was an amino acid with like a hydrophobic R group and another amino acid that's substituted with a hydrophobic R group, shape of the protein might not change that much, so it might be fine, right? The third category we call nonsense, because a nonsense mutation causes a premature stop. Codon in the message. Right? So all the way back to the beginning, there's a change to the DNA sequence that changes the messenger RNA codons, and a codon, a messenger RNA codon, one of the codons becomes a stop. And so it's premature because it's not at the end, somewhere before the official stop codon. And then there's a whole other category of mutations altogether that are not these are examples of different types of base substitutions, remember, or what we would call base substitutions. There's a whole other category of point mutations that we call frame shift mutations. And a frame shift mutation is a point mutation, so it happens at one place, one point. In a frame shift mutation, we have base or bases inserted or deleted in the DNA. That's going to shift the reading frame, which is why it's called this. If you can remember that, that shifts the reading frame. It shifts the sets of codons in the messenger RNA. And the protein is a mess. That dramatically changes the protein. It is usually not functional, especially if it's an enzyme. Now, the main way we can detect if a substance is a mutagen is by doing a simple test in bacteria. It's called the Ames test. And it's really important because scientists actually in pharmacology that work at pharmaceutical companies actually still are required to report the results of this test as part of their toxicology findings when they are trying to put a new drug to market. Okay, and that test for mutations is called the Ames test. So I'm going to erase it again, but it still kind of comes under the big category of mutagens or mutations. So, Ames test. This is a test we can do in bacteria to determine if a chemical is a mutagen or if it's probably a mutagen. Because the test goes like this: if it's a mutagen in bacteria, it is probably 80 to 95% a mutagen in animals, and that usually leads to tumors or cancer. Okay? So I'm going to go through the Ames test real quickly here, just to show you how it's done. And I'm going to show you, we're going to do an experimental test tube and a control test tube. And into each of these test tubes, we're going to put some media, some broth, so that the bacteria will grow, because that's their food, remember. And always in the Ames test, we always use the same bacteria. So the scientists can purchase this bacteria from a company. It's called the American Type Tissue Culture Collection. So everyone's using the same bacteria. And it is a Salmonella genus, Salmonella. And sort of superscript we put up here, His minus, because it is a Salmonella that cannot make histidine. So it's already mutated. So you'll see it like written like this, SAL for Salmonella and the superscript histidine minus. Histidine is one of the 20 amino acids that bacteria have to use to make their proteins. Okay, so what we're going to do is into both of these tubes, I'm going to make this first one the experimental and this one the control. This is a negative control. I'm going to add my bacteria, Salmonella His minus. Salmonella His minus. Whoops. I'm going to add that to both. To my experimental, I'm going to add chemical X. All right. I know you guys are too old. You probably didn't watch the Powerpuff Girls, but my kids did. So to this one, we're going to add chemical X. And then, of course, to the control, to the negative control, we're not going to add chemical X. This is our suspected mutagen, chemical X. Right. And then you also need to add some rat liver extract. Don't ask me. Makes it work better or something. Then you're going to plate the bacteria on a petri dish, and the media that you make lacks histidine. And you actually use two plates, one with histidine and one without, but we're just going to look at the His minus plate. Okay, this bacteria cannot make histidine. If I don't supply it as like a vitamin or as a supplement in the media, the bacteria, if it's mutant, won't be able to grow, right? Or if it's wild type, I should say, it won't be able to grow. However, in our positive control, our negative control rather, there are always a few colonies of our Salmonella His minus that we say reverted to His plus. Now, how is that possible? Come to genetics, you can learn more about it. What you just need to believe me that this mutation, the reason that everybody uses the same Salmonella His minus for the Ames test is because this particular histidine mutation in the DNA is at what we call a mutational hotspot, which means just like it sounds. The DNA mutates to that, but it also can revert or mutate again. Remember, every time the bacteria multiplies, we have the opportunity for a spontaneous mutation. So this is actually, this Salmonella His minus, the histidine mutation is at a hotspot. So there's always going to be a few, less than 10, colonies that are just spontaneously going to revert back to His plus. So they can grow on this plate without histidine being added as a supplement. They can just make it, we say, from the minimum concentration of things, from minimal media. Now, what we're more interested in is how many colonies grow on this plate. Because in my example, any number of colonies over 4 is going to indicate that there are more revertants, which means there's a higher percentage chance, higher percent of mutations caused by chemical X. And like I said, this is super simple. It's a bacteria. It's going to cost you $50 to do this test, not hundreds of thousands to do the animal test. So if something is positive by the Ames test, okay, don't put it in food, don't put it in a medication, because most likely it's going to be a mutagen in animal cells, and if it's a mutagen, that means it's a carcinogen, right? So this test is super important. It is done to everything that's going to be added to food, any food additive, a food coloring, a new compound, even if it's a naturally occurring compound. It has to pass the Ames test before it can be put into food by so the FDA knows that it's safe. Same for any medication or pharmaceutical, has to be tested by the Ames test before the FDA approves that it can be put into a particular medication or a drug. And that, my friends, is the end of chapter 9, finally. So I'm gonna stop the recording.